Hi class, in this recording we're going to focus on the interior anatomy or internal anatomy of the spinal cord. So first, let's dive in and look at some of our cross-sectional anatomy. As we're taking a look at this, let me narrow that down a little bit here. Our spinal cord has two different colors inside of it. We have gray matter. Let me switch here. There we go. We have our gray matter and we have our white matter. Now, if we look at the inside of the spinal cord, the gray matter is going to be deep in the spinal cord. And this is different from the brain. Within the brain, the gray matter is going to be on the surface of the cerebrum as opposed to white matter, which is on the surface of the spinal cord. So the gray matter, which is deeper towards the inside of our spinal cord, is going to be consisting of primarily of the cell bodies of neurons, also known as neurosoma. Cell bodies of neurons are not myelinated. Because they are not myelinated, they don't have a lot of fat in this area that within the gray matter, and we have the color gray. It's worth emphasizing that there are lots of dendrites present. These dendrites that are present within the gray matter and the neurosoma themselves allow for information processing. We have lots of synaptic integration. And if you don't remember, a synapse is a connection between two adjacent electrically active cells, in this context, two neurons. The white matter is superficial to the spinal cord and is located on the surface. This white matter is made of abundantly myelinated axons. And when we say abundantly myelinated, we mean that there's lots of electrical insulation around the axons that make up this tissue. And because myelin is high in fat or has a lot of lipids in it, the white matter has a white coloration. When we think of white matter, I want you to think of movement or information that, or the tissue that carries information from point A to point B primarily. And when we think of the gray matter, I want you to think of the tissue which processes the information primarily. Now, as we're looking at gray matter within our spinal cord, the gray matter can be broken up into a couple regions. We're going to be focusing on one half of the butterfly down here. So first we have the posterior horn, and I'm a big fan of the posterior horn and that terminology because it's towards the back. If you're a little older like I am and you're getting crusty, you may have been taught dorsal horn, which is a term that works really well for quadruped animals, but we as humans are bipeds, and that term is starting to be phased out just because of me kind of confusing. As we're looking at the ventral horn on the front, aka the anterior horn, that's another region of the butterfly. And then finally, there's this bulge out to the side, and when we think side, think lateral, that is the lateral horn. And then connecting the left and right sides of the butterfly or the gray matter, we have this tiny little branch. That tiny little branch is referred to as the gray commissure. And then in the very center of the gray commissure, there's a small opening. And that small opening is quite large when we're younger. As we age, it fills in. It's known as the central canal. Or I should say central canal of the spinal cord, to be more precise with my language. And the central canal serves as a site for cerebral spinal fluid to circulate throughout. The central canal is going to be lined with ependymal cells. Last chapter, in chapter 12, we talked about ependymal cells. They are one of the neuroglia that are present in the central nervous system. Specifically, the ependymal cells have cilia on their surface, on their apical surface, which move the cerebral spinal fluid that they produce. As we look at the lateral horn over here, that lateral horn is only going to be present from the second thoracic vertebrae to the first lumbar vertebrae, those regions of the spinal cord. So up in the cervical regions, and for most of the cervical regions and the very beginning of the thoracic regions, there is no lateral horn. As we look at that lateral horn, it contains neurons of the sympathetic nervous system. And as we think of the sympathetic nervous system, I want you to think of the nervous system responsible for fight or flight responses, our high anxiety, high stress responses. It's a subdivision of the autonomic nervous system. As we look at the white matter is going to be superficial to the spinal cord, and it's going to be broken up into columns, sometimes referred to as funiculi. I'm much more of a column guy personally because I see these regions of white matter that go up and down like a column. I'm not quite old school enough to have been taught funiculi and have that be the way I think about it intuitively in my brain. And as we look at these columns or funiculi of white matter, that we have three big chunks. We have, right now that I'm presently circling, the anterior, we have the lateral column, and then we have that posterior column. And within the columns, we have tracts. These tracts are going to be small subdivisions, so we can take one column and break it up into different areas. And those different areas, I'm using yellow, blue, and red right now just to show that one column can be broken up into different areas or subdivisions. So when we think of a tract, this is a term that I haven't really spent a lot of time defining. You can think of a tract as a bundle of axons in the central nervous system, a bundle of axons in the central nervous system. If it's in the peripheral nervous system, we call it a nerve. Here we have a spinal nerve, a bundle of axons in the peripheral nervous system. Here we have a bundle of axons in the central nervous system. We call that a tract. And as we're looking at the tracts, some tracts are going to carry information up to the brain, some are going to carry information down from the brain, and some are going to cross over, some don't cross over. There's some interesting things these tracts can do. All the fibers in a tract, to differentiate one tract from another tract, we have to look at the axons or the nerve fibers that make them up. They all have a similar origin, destination, and function. So as we look at ascending tracts, this term ascending refers to the fact that ascending tracts carry information up, particularly if it's going to the brain. It's going to be sensory information going to the brain. And then the brain integrates or processes that information, makes a decision, and then through the descending tracts carries motor information down. Now, something that can happen with these tracts is there can be some decussation. And this decussation is when we have some crossing over. In particular, we can have the tract cross over at the midline. And this allows for the brain to control a contralateral side of the body. And when we say contralateral, don't worry, I have to find the word right there. This is a way of saying that the origin and destination tract are on opposite sides of the body. So for instance, if you're looking at somebody and can't move their left arm, that's a distinct clue that they may have had a stroke on their right motor cortex, or the right primary somatosense, right primary motor cortex. The right motor cortex controls the left side of the body. The left motor cortex controls the right side of the body. They are contralateral. There is that decussation because they cross over to opposite sides of the body. There's also going to be some ipsilateral. And when we think of ipsilateral, there is no decussation, and there is no crossing over of the tract. So it starts on the right side of the body and ends on the right side of the body, or it starts on the left, ends on the left. So this is what I was referring to with the ascending tracts going up, descending tracts going down. If you are one of those individuals, those intrepid students that are reading their textbook, your textbook goes into a lot of detail about all of these different tracts within the spinal column. I don't think we have time in this class to cover them. Class in neuroanatomy, and you can learn about those individual tracts within the ascending and descending columns in more detail. What I really want you to focus on is the big picture idea that ascending tracts carry sensory information up, descending tracts carry motor information down, and if they decussate, they cross over. And if they're because they are contralateral, and if they're ipsilateral, they do not cross over. They stay on the same side. Now, as we're looking at our ascending tracts, just to emphasize again, they carry sensory information. And typically, there's going to be 3 total neurons involved to get that sensory information processed. Our first order neuron is going to take that stimulus, the heat, the cold, the touch, the pressure, whatever it is, take that stimulus, convert it to an action potential, and then transmit that to the spinal cord or brainstem. Our second order neuron, number 2, is going to start at the thalamus. Or excuse me, it will go from the brainstem to the thalamus. And then finally, our third order neuron is going to go from the thalamus up to the sensory cortex, sometimes referred to as the accessory sensory cortex or the primary somatosensory cortex, the sensory region of the brain. So step one, converts the signal and carries it. Step two, moves it around within the brain a little bit. Step three, we take it to the point of processing. As we're looking at those descending tracts that carry signals, motor signals, so signals from muscles and glands, down or inferiorly from the brainstem and spinal cord, there's typically going to be two neurons. The upper motor neuron is going to start in the motor cortex or cerebral cortex. Occasionally, it starts in the brainstem, depending on if we're looking at something that has voluntary versus involuntary control. And then that upper motor neuron typically is going to go all the way the length of the spinal cord and terminate at the lower motor neuron. The lower motor neuron is going to have its neurosoma either within the brainstem or within the spinal cord, depending on where it's located in the function. For me personally, I always like to diagram it out and think of it as the lower motor neuron having its cell body of the neuron or neurosoma within the spinal cord. So we have starting in the brain, going to the spinal cord, and then we start at the spinal cord and then go to the target organ. So this axon of the lower motor neuron is actually going to exit the central nervous system and go into the peripheral nervous system to hit its target organ. As we're looking at the corticospinal tracts are just going to be regions that carry signals. And so we have that term again, tract. So when we think of a tract, think of a bundle of axons within the central nervous system. Corticospinal tracts are going to carry information from the cerebral cortex, allow for us to have very precise, finely coordinated motor movements. And as we're looking at these corticospinal tracts, they are going to form some pyramids, which are some bulges right here that are going to be these anterior bulges on the medulla oblongata, where we have the medulla oblongata in kind of salmon. We have the pons in blue, and then we have the midbrain in green. And as we're looking at the tracts that are within these pyramids, they descussate in the lower medulla. So here's the lower medulla where we have that site of descussation or crossing over. Let me clear the screen here. And because they desiccate, they are going to form lateral corticospinal tracts on the contralateral side of the spinal cord. So in other words, when we think of contralateral, think opposite sides. So left versus left to right, right to left. Sometimes they will be descussation lower down in the spinal column, but this site right here is where we begin a lot of our descussation or crossing over of the tracts from left to right or vice versa. And that's all we have for this recording. If you have any questions for me, please feel free to post them on the class discussion boards, shoot me an email, or as always, and always, have happy studies.